Good morning and welcome to the IFAN's Bioactive Committee-sponsored webinar on a research and application-driven cloud resource for precision nutrition. I'm Janet Novotny. I'm a research physiologist at the USDA Beltsville Human Nutrition Research Center in Beltsville, Maryland, and I'm the government liaison to the IFAN's Bioactives Committee. We're so glad that you're here and joining us for our webinar today. This slide shows my disclosures. Before we start the talks, just a brief background on who is IFANS, the Institute for the Advancement of Food and Nutrition Science. IFANS mobilizes scientists from government, industry, and academia to drive actionable research in food safety and in nutrition. All work is collaborative in nature and advances public health. The website is a great place to start if you're interested in learning more about the programs and the content. Attendees are invited to submit questions throughout the webinar, and the questions will be discussed after the presentations are made. Please write your questions in the Q&A section rather than the chat section. Our webinar today will include 5 speakers, starting with Colin Kay, an associate professor at the Plants for Human Health Institute at the North Carolina State University, Sierra Convey, a PhD student in the Department of Bioinformatics and Genomics at the University of North Carolina at Charlotte, Alexander Smirnov, research assistant professor in Bioinformatics and Genomics at the University of North Carolina, Jean Yang, professor in the Department of Computer Science at the University of North Carolina, and Shusha Du is a professor in the Department of Bioinformatics and Genomics at the University of North Carolina in Charlotte. We will now have a brief pause while we switch to the presentations.
Good morning, everyone. Thank you for taking the time to be with us today. Today, our team is going to describe our recent initiative to establish a cloud resource for precision nutrition. Here's my disclosure slide. So the first part of the talk, I will be outlining common food and nutrition databases, database harmonization, the importance of knowledge databases, precision nutrition, led by Metabolomics and big data analytics, and then our efforts to develop a precision nutrition knowledge database. So the value of nutritional databases is quite apparent to most of us. They provide details on diet patterns and food composition. They support practitioners and researchers, but we must admit there are a number of challenges and areas of improvement in this respect. So when we think of the common databases in nutrition, we often think of the USDA databases or the NIH ODS databases, or some of the more search engine type databases like Phenol Explorer, PhytoHub, or FoodB. We can think of these as a collection of data in single Excel files, or perhaps linked data sets using software like Access, or perhaps an online search engine type database. Really what we're moving towards is network databases. So when we think about the USDA database, we often think of the survey data, like what we eat in America. When we're on this website, we can click on this link and we'll get a document that gives us additional information on how the data was collected or how to use the database. Some users might want to click on and get a PDF file, which is providing a card of compositional data, and others who have the know-how would be able to download an Excel file or a data set and incorporate that into their workflow. Similarly, for USDA Food Data Central, we have simple searches, or we can click on something like Foundation Foods, basically to get a list of downloadable data sets or workbooks. When we click further, we might get a file that we can unzip and a series of linked data sets. And beyond that, you can see we can get a unique data set which we can export to Excel or Access or whatever database software we're using. And not to leave anybody out, here's the beautifully reworked Office of Dietary Supplements databases. And similarly, we can click on a data set of interest. We may be able to download a research summary as a PDF, or we may be able to download a data set, which you can see is quite large, up to 6,000 or 7,000 rows of data commonly. And the important thing to note here is with many of these USDA and NIH data sets, we've got common data elements like the DSID or the NHANES identifiers, which basically allow us to, when we're looking at multiple data sets within these databases, link the data sets using these common data elements. So these are all valuable resources that we all use throughout our workflows, but I call these single click in, click out interfaces. So you click on a door, you go through the door, you get some data, you click out, you click into another door, get some more data, click back out. Now, wouldn't it be wonderful if we had a way where all of these data sets could communicate? So basically what we're talking about is networked databases or network data sets, which is where we're all moving towards in the future. In proteomics and metabolomics, they're a lot further along than, say, we are in some of our traditional nutrition databases. So what we need to consider is something like a launch pad or a dashboard, which would allow us to see all of our data in one place. Data visualization is a fantastic tool which allows us to do this. We can still embed simple searching strategies as we're all used to using, or a system which we're implementing which involves both essentially. The key is that we need to be able to drill down to the source data. We need to be able to identify the references of where the data comes from, download identifiers such as database IDs, or ideally even link directly to databases where we can search for additional information. So what's needed in moving forward? Well, first we need to understand how are we presently utilizing these databases, and we need to think about all the user groups that use these for precision nutrition. We need to think about experts in metabolomics and exposomics, or in nutrition, maybe people involved with cohort studies or nutritional intervention studies, and not forgetting actual people. This is precision nutrition. We need to drill down to the people in the public that would potentially use these resources to inform their diet and lifestyle. So who is using these databases and what are they using them for? Well, it may be practitioners or lay persons that are looking for information or maybe for menu planning. We also have to consider people in marketing and people in regulation that may be using these for labeling or informing policy. And we also need to consider researchers. So researchers in nutrition or food science or exposomics all use these various databases within their workflows. Now, in moving forward, then we have to understand the workflows of the potential users. So if you think of the metabolomics workflow, it's quite involved. And if we consider it maybe a top-down approach, we have a number of instruments being used. We have a number of annotation softwares and analytics softwares. And then we communicate with a number of databases to find more information about compound structure, say, or where these compounds may be within food, or where they may be within the diet. And then we can move on to pathway analysis, and there's various online resources and databases to look at pathways, and ultimately making the link to health outcome. So you can see there's a number of databases and knowledge bases used within this workflow. If we consider more the nutrition focus, maybe personalized nutrition workflow, it's kind of coming at it from the opposite direction. So we may use the USDA or NIH databases to generate some understanding of diet. And then we may link that to food, and then we may link to compositional databases. And then next we may want to establish what metabolites are known relative to those compositional items. Then we might move to metabolomics databases to establish, have others identified these compounds? What studies are reporting these compounds of interest? And then again, we can move on to pathway analysis and making links to health outcomes. So there are a number of limitations for users that we need to consider. So there's really limited capacity to integrate data sets and databases with each other. There are internal identifiers which are great, but these databases are often lacking common data elements required to link to other external databases. So some of the challenges moving forward is we can all agree that many of these interfaces are very tedious to navigate. And we need to consider that user habits evolve. So consider the younger generations who have grown up in this app-based society. They're not going to click 3 times to get a document on how to use a resource. If they're not getting data after the first click, they're moving on. So we need to consider all the users. We also need to consider one of the challenges is staying up to date with data, obviously. And then with the breadth of data and abundance of data that we have, we need to be concerned about data quality and data quantity. So what is a knowledge database? Well, a knowledge database is essentially a highly dynamic cross-link database or cross-linking of data sets. So the benefits of this type of database is that can provide a one-stop shop that will help people more easily define gaps in knowledge. They can be used for both hypothesis generation and hypothesis testing, which is very useful. They're highly interactive, and they can use data visualization techniques and machine learning. Now, the challenges, as you can imagine, are that we need to be concerned with data quality. We also need to be cautious that high level of complexity can't be in the face of usability. So we still need to make sure people can navigate these systems. And there's costs associated with data storage and maintenance of these knowledge bases. So how do we then move from these static databases, which we see a lot in nutrition, to more knowledge databases? And that's really, we need common data elements. Um, so if we think of a common data element as just being a linked identifier, so in the USDA and the NIH databases, it's relatively easy to use their common identifiers to go from, say, a diet to a food ingredient, but it's maybe not so easy then to go to a molecule within the composition or a metabolite or a metabolic pathway or a health outcome. And there are databases which focus on all of these things and they share common data elements. So when we start sharing and pulling from multiple databases, though, we need systems in place to ensure data integrity. So we need systems for data harmonization, systems for establishing quality of the data. We don't want to propagate error, and we need to be able to identify discrepancies between databases. And ultimately, then we need systems for reporting data assuredness. So what precision nutrition databases are presently available that we can maybe learn from? Um, Well, if we consider all the user groups I've outlined in this diagram here on the right, I haven't found one yet. So if anyone knows of any that satisfy all these user groups and the present definition of what we consider to be precision nutrition, put it in the chat. I'd like to see it. Basically, what I've been able to find is usually these are private databases calling themselves precision nutrition databases. They allow you to search, generate ingredients lists or meal plans or exercise plans. They're highly advertisement laden, so there's an element of public distrust when they click on data sets or databases like that. And often they're trying to sell you a plan. Um, so what are the obstacles in moving forward? Phytochemicals are poorly represented in compositional databases. And obviously we consume a large number of phytochemicals. Nutrients are better represented, I would say, at this day and age. We've also found that metabolites are poorly characterized or captured in existing databases. Phytochemical taxonomy and nomenclature is poorly defined and inconsistent. And really the data is scattered across data sets and databases, which often lack these common data elements. So in our initiative, we started with these two fundamental questions, and that is, what is the contribution of dietary phytochemicals to the human metabolism or metabolome, and how does their contribution affect health and wellness? And really, we wanted the fastest way to bridge question one and question two. So we felt that that was through establishing our own in-house database, which we are calling PMetDB, or Phytochemical Metabolome Database. We needed to be able to integrate with Metabolomics databases and software, and ultimately form a knowledge database, which we're calling the Metabolome of Food Knowledge Database, which allows us to build in data visualization, which allows us to make this more accessible to researchers and the public. The purpose when we initially started was we felt that the fastest way to link diet to health would be to give people a tool that would allow them to identify the contribution of diet to metabolomics dark matter. So we can go through previously existing data sets and form some better associations between diet and health. We started with an evidence mapping initiative. Again, we're calling PMetDB. We needed to start by considering what phytochemicals we should be focusing on and where are they in the diets. We need to focus on bioavailability of things, not just what's in food, the metabolites when we eat those foods, and what are the analytical properties of those analytes. And then we needed to think about biological activity. Do we go to the literature to decipher all of the published data on activity, or move to databases, which are a little easier, obviously, to curate than doing hundreds or thousands of literature searches? So the first step is the curation step. So that's really the acquire of data, clean the data, and establish compound and metabolite identifiers. So our pathway for development of this database was to first start with target identification using NHANES and USDA databases, then conduct systematic reviews using PubMed and Web of Science. Then we extract data for phytochemical composition and also metabolites. And then we look for chemical identifiers which match that compositional data, things like InchiKey or formula or accurate mass, also capturing database identifiers. Then we needed to establish a hierarchical taxonomy for chemicals and reactions and pathways. Then we moved on to building a reference standard library, so when we do find things in our research, we can prove their existence or their identity within our metabolomics data sets. We also modeled metabolites based on known microbial on phase 2 conjugation. Then the next step is to build our MSMS and HRMS reference spectral libraries. Then we moved on to our disease pathways, and then ultimately our data visualization, which is presently involving a self-organizing map, Sankey diagram, and node link diagram. And these steps will be described in further a little later on in this webinar. So for the curation and cleaning stages, we initially identified about 13,000 manuscripts and identified about 900 which contain compositional data of plant composition or metabolite identification in human samples. We have various expansions underway, but initially we focused on kind of 10 of the most consumed phytochemical dense sources in our diets. So you can see the list here. We initially extracted anywhere from about 150 to 2,500 lines of chemical composition data. And this translates anywhere between 30 to maybe 500 analytes after we remove duplicate data. So moving forward from our PMetDB to our Metabolome of Food Knowledge Database, we needed to consider food, metabolites, pathways, and diseases. So we needed a way to group data. So we needed to establish a hierarchical phytochemical taxonomy. which essentially allows for various levels of grouping or clustering in our data visualization interface. So we wanted to focus on bioavailability as opposed to just quantitative abundance in foods, because we need to consider that is it really impossible to have a consistent, up-to-date, real-time, quantitatively accurate representation of food composition? If we consider the chemical composition of a plant food, what we observe is going to be dependent on the number of replicate observations across a number of years, a number of growing conditions, processing conditions, and food preparation conditions. We also need to consider that the food supply chain is constantly evolving. So we have evolving consumer preferences, we have hidden ingredients, and we got changes to the supply chain or the cost of ingredients, which will change how a product is formulated. Consider spice mixtures or oil mixtures. Often they're not listed, or they're a mixture of various oils, which will vary depending on the availability of certain ingredients. Also, we need to consider biology over static food quantity. So if we take this arbitrary food in the first diagram here, let's say it has only 10 analytes in it. Obviously, that's an oversimplification. Here's the concentrations of those analytes. If we then consider the bioaccessible qualities, so that's the level that can be extracted from that food matrix and made available to absorption and metabolism, this graph looks very different than the top graph. And then let's also consider at the bottom bioavailable quantity. So this is the level that is absorbed and we see in the blood. We'll see a very different concentration of these analytes relative to what's in the food. We also need to consider phytochemical metabolism. Let's just take a made-up molecule. It's a polyphenol consisting of 2 rings. Our microbiome can break this down, say, to 4 separate analytes, when in fact we know it's often quite a higher number. And then our human phase 2 enzymes can conjugate these structures to, say, 4 other conjugates. So what we see in the human metabolome or the blood is that for every molecule of the precursor found in the food, we could have 4 microbial metabolites and 16 hybrid human microbial metabolites. And lastly, we need to consider the differences in metabolism across people. If we take that same hypothetical molecule and let's say it has 10 metabolites, and these graphs represent 10 different people's metabolomes, what we see often in our studies is that nobody metabolizes these compounds to the same extent. Some will be higher for one analyte and lower for another. So what we're seeing is that what's in the blood has very little representation of what's in the food. So the focus on static quantity in food, we don't think is the appropriate way forward for precision nutrition databases. So then moving forward from the food to the metabolite, we need to move to the disease or pathway. So we also need to consider the complexity of moving from a reaction to a pathway. So in a reaction, we have kind of a precursor structure and a product structure, and then we have a pathway which will be connected to a neighboring pathway. And in fact, pathways are highly networked, so it's much more complicated than this linear relationship. So in this case, we need to consider various levels. We need to consider the compounds in the foods or our metabolomes. We need to consider the reactions they participate in, the metabolic pathways and biological pathways that they regulate. We need to consider metabolic regulatory systems, genetic regulatory systems, biological systems, so these are tissue and organ level systems, and system pathology that may link these tissues and organs to disease pathologies. So we need to come up with a hierarchical way of ranking these associations, and this will be described in further detail a little later in the webinar. So we've essentially established a cloud resource. We're calling it MetaboFood KDB for Knowledge Database. It allows people to visualize the uniqueness and overlap of foods using a Sankey, a self-organizing map, and a node link. It allows people to interactively explore linkages between food, their chemical compositions, pathways, and diseases. and it allows us to search for compounds and metabolites associated with food and disease. So the Sankey is shown here. You can see it's more complicated and it has more utility for research. We have a self-organizing map, which is taking three-dimensional data and really turning it into one-dimensional data in a very visual manner, which is a lot of public utility. And then we have our node link map, which allows us to interact with our nodes in a highly refined manner. And again, we'll present a little more on that a little later in the webinar. So essentially, we're building something that focuses our feature development based on our users. So our primary goal is making big data publicly available, but that doesn't mean only for the science savvy. So we want it to be available to the general public. So we're embedding features which allow things to be accessible by the lay public, but also have utility for nutrition and metabolomics researchers. So we're initially designing our development based on 5 user groups. So we have kind of the level 1 is our personalized nutrition, our nutrition or health curious, and also it would have utility for nutritionists, nurses, and dietitians. Level 2 is kind of more advanced. We have precision nutrition researchers in epidemiology or intervention studies. So they might use this to identify diet pattern clustering or establishing food questionnaires or intervention study design. Then we have our untargeted metabolomics, our biomarker discovery, and our exposome researchers as level 3, which would allow you to drill down to identify unknowns and establish confounders in data sets. And not to leave anybody out, we also want to have data that's useful for people in medicine and drug and toxicology research. And lastly, in microbiome research, as a number of our metabolites captured in our database are microbial in origin. So our focus and future development is to provide the public with the ability to input dietary items and see how these map to health status, and to make dietary alterations which support disease prevention strategies. Also to provide researchers the capacity to upload data or diet components and explore associations with metabolomics data sets, biochemical and disease pathways. And finally, to provide the opportunity to design precision nutrition diets for personal or nutrition research use. So I'm going to stop there, briefly introducing our team. You're going to hear next from Dr. Shishadu, Dr. Jin Yang, Dr. Alex Smirnoff, and one of our PhD students, Sierra Connolly. So thank you for your time, and I will hand this off to the next speaker.
Good morning. My name is Sierra Conway, and I am a second-year PhD student in the Do Lab at UNC Charlotte. Today I'm going to be talking about the efforts that went into collecting the necessary information to extrapolate upon the phytochemical data curated by Dr. Colin Kay's lab. And specifically, I'm going to go into details pertaining to the process and databases that were used to establish the different relationships that were ultimately implemented within MetaboFood KDB's interface. So the purpose of these efforts was to develop a workflow that could connect varying facets of metabolomics information from these multiple databases. For MetaboFood's KDB, we are interested in connecting the metabolite to its respective pathways, and then once we obtain those connections, we look at how these pathways could be implicated or related to a specific disease or disease progression. And since the accuracy and robustness of these connections for these hand-curated phytochemicals kind of establish the backbone for MetaboFood KDB, it becomes imperative to develop a solid workflow that could effectively data mine and connect these multifaceted bits of information. In addition, that information collected has to provide the user with a thorough, accurate, and meaningful insight. So here is the general schematic of the process that was used in finding and harvesting all this data. So for easy visualization, I've separated the process into 3 distinct parts. You can see to the right on the flowchart. The first is creating the metabolite to metabolite relationship between 2 different databases. In this case, HMDB to KEGG, and then from KEGG to Reactome. So I take that external ID from the raw data that was given to us. In this case, it's HMDB, and associate it with its respective KEGG ID. if it exists. And in some cases, we didn't have an existing KEGG ID, and we handled those compounds differently. Same goes for KEGG ID to reactome ID. We collected those that did have reactome IDs, and those that didn't, we handled them differently. The second part is taking these metabolite IDs and establishing the metabolite to pathway relationship. This was completed in two ways. One was through KEGG, and we collected the KEGG pathway IDs that had been associated with those KEGG compound IDs previously found. And then the other was through reactome, where the reactome metabolite IDs were then matched to their respective reactome pathway IDs. And the last part was taking the pathway IDs from both KEGG and reactome and matching them to disease IDs from CTD, which is the Comparative Toxicogenomics Database, which allowed for the pathway to disease relationship to be established. So I quickly wanted to touch on the databases that were used. So you have HMDB, KEGG, reactome, and CTD. HMDB to KEGG is that metabolite to metabolite relationship. KEGG is also pathway, and reactome is metabolite to pathway as well. And CTD is that pathway to disease database that was used. And below is a description of kind of the purpose of each database and what it does. So I wanted to talk about each step in more detail. The first step is that metabolite to metabolite relationship that was discussed previously, which was the HMDB to KEGG conversion. So a Python script was written to parse the HMDB database and then merge that information into the original spreadsheet. So in addition to collecting KEGG IDs, I also wanted to collect any external IDs such as Drug Bank, PubChem, Metlin, to give some more robustness to the to each phytochemical. And so in the future, if we wanted to, kind of explore those databases, we could. We'd have an identifier to them connected to the compound that was given to us. I also collected structural information such as SMILES, INCHI, and INCHI-key, which is used later in the talk. And so you can kind of see from this figure below, you can see the HMDB ID and then its related KEGG ID in the spreadsheet. So for this next part, previously it was a HMDB to KEGG. So this is the KEGG to reactome ID relationship that was formed. So that was done by using the API call within Python. And first you have to find that database ID. And then secondly, you find something called the stable identifier, so the STID. That stable identifier, you can have multiple stable identifiers for a single compound. It essentially is what is this compound like under certain conditions in the human body? And that is how we kind of related next to the pathway IDs. You need that stable identifier. So that brings me to my next part, which is the metabolite to pathway relationship. So in KEGG, what I did was we used the Python best KEGG API call to obtain those KEGG pathway IDs from the KEGG IDs, and this was in the form of a JSON file. And you can kind of see here, in this case, I have carafilene, beta carafilene as my example. You can see the HMDB, then its related KEGG, and finally its Homo sapien related pathway. And if you go to the website and to the webpage, you can actually see how these bits of information are kind of connected together visually. So you have the KEGG ID and then the map, and then which leads to the pathway that you can see and visualize. So in addition to collecting the KEGG pathway IDs, I also went in and collected the reactome pathway IDs. And that was done through the reactome API call, which was used to obtain those reactome pathway IDs from the stable identifier. And then again, in the form of a JSON, which I just parsed and matched to its respective phytochemicals. So an example would be homovinylic acid, which is the last step in dopamine metabolism. And you can see here, you've got the KEGG to database ID to the last two, which are in red, which is the stable ID and the pathway ID. And you can kind of see on the webpage how that stable ID is related to this particular pathway. In this case, it's enzymatic degradation of dopamine by a particular gene, COMT. And you can actually visualize it within Reactome. And what's really cool is, in addition to the pathway IDs, I pulled these PNG files of every pathway we had that related back to any phytochemicals. So that gave us another layer of visualization. So finally, you have that pathway to disease relationship. And this was both for KEGG and Reactome. In this case, you can kind of see here with CTD, you have that pathway ID, which related to a disease ID, which is then related to a disease name. And in this case, you can kind of go to the CTD and in search, and you can see here the corresponding disease in web page form. So once all the information from each relationship was collected, we kind of needed to find a way to group both pathways and diseases in an effort to kind of declutter the data. Since Reactome and CTD have built-in hierarchies already within their database, I was able to match parent pathways or diseases to their respective child. It is important to note that for the CTD, we utilized only the top layer of the hierarchy for convenience. And this kind of created a more organized visualization of the information. So now I kind of want to get into the additional information we gathered and what we did with those compounds that did not have an HMDB or external ID to match to. So, and that's where the smiles came in. And the importance of that is that smiles can be used when using RDKit. So I did this within RDKit. Can be used to visualize the compound. You can also use something called the Tanimoto similarity and actually cluster compounds based off their smiles. And specifically, you go from smiles to fingerprints, and then you cluster those fingerprints. And so from there, those compounds that did not have an identifier, we were actually able to cluster them together. And you can kind of see in this example here, you have similar compounds that where some have a KEGG identifier and some don't. And it kind of gives us a kind of visualization into how those compounds that don't have a whole lot of information might actually fit into some compounds that are more well known. And so that process was kind of done with finding those valid INCHI keys and through the external identifiers. And if there were no external identifiers, I wrote a script that would take the INCHI key and find the INCHI in its respective smiles. So I kind of going into future works, I kind of think it would be great to expand the smiles-based algorithm to cluster those compounds that have related KEGG IDs to those that may not, or compounds that we're just beginning to discover that wouldn't be in a database like KEGG or HMDB or Reactome. And so it'd be nice to kind of further expand on that and also cluster based off of binding affinity and kind of see which compounds actually could function similarly, not just have a similar structure. Secondly, it'd be nice to mark those compounds that are identified as biomarkers. So as you can see to the right, I've actually already done this through MarkerDB. And it would be nice to just kind of have those within the interface. So those that are biomarkers are also identified as such. And thirdly, it'd be nice to use the software to attempt to predict metabolite metabolisms, specifically for those compounds that are not well known. And we could potentially do this through biotransformer. So I want to thank the Do Lab, the K Lab, and those other individuals that helped me to further this project along, and I'll be happy to take any questions.
In my part of this webinar, I'd like to present our current work on the MetaboFood web application. This is the conflict of interest disclosure and all the grants that our lab is currently working on. When we started working on the MetaboFood, we had several goals in mind. First, Dr. Colin Kay spent several years on creating and curating a comprehensive dataset of metabolites and their relations to common food types. However, in order to use the dataset, there was a need in a user-friendly interface that would provide an easy way to search for and retrieve information. That's why we started working on the MetaboFood web application. Second, we want to provide MetaboFood with easy-to-understand visualizations that can be useful not only for specialists, but also for common users. And finally, we want to help users to explore relations between foods, compounds, pathways, and diseases, and make the MetaboFood interface and its visualizations highly interactive and easy to use. From a technical perspective, we are using open source technologies for MetaboFood that are free to use for both commercial and non-commercial purposes. SQLite is a small database engine that is very easy to set up and maintain. For our web development, we prefer using the Flask web framework because it's written in Python, which makes it easy to incorporate any machine learning algorithms if needed, through popular Python packages such as scikit-learn and TensorFlow. Finally, Dr. Jin Yang has a great experience of developing dynamic interactive visualizations using the D3 JavaScript library. On this slide, you can see the current structure of the MetaboFood database. The main database entities here are the food, compound, compound taxonomy, pathway, and disease. The compound taxonomy includes the family, class, subclass group, subclass, and metabolite. All these entities are interconnected and can be easily accessed from the MetaboFood web application. Next, I'll go over the functionality that is currently implemented in the MetaboFood. First, when users are interested in specific food type, family, or class of the compounds, specific pathways, or disease, they can search for whatever they need on our homepage and start the exploration from there. From any compound, pathway, or disease page, users should be able to explore the taxonomy of compounds. Currently, the compound taxonomy includes the family, class, subclass group, subclass, and metabolite. Users will also be able to explore the hierarchies of pathways and diseases. We'll provide an easy interface to explore relationships between foods, compounds, pathways, and disease. I'll present a Sankey diagram that we use to visualize the relationships. We'll also provide an interface to evaluate similarities and differences between different food types with respect to their compound compositions and relations through pathways and diseases. Dr. Jin Yang will talk about this later. On the left figure, you can see our homepage. Currently, it only contains a search box. Here, users will be able to search our database records for a specific food, compound name, compound class, compound subclass, pathway name, or disease name. After users click on the search button will display the matching results that can be further sorted and filtered. You can see the search example on the right. From here, users will be able to click on a specific food type, compound, pathway, or disease to display detailed information about them. When users click on a compound family, class, or subclass, will display the taxonomy page. Here, users can browse all the compounds contained in that family, class, or subclass, and again sort and filter those compounds. In addition, we display the taxonomy diagram where users can browse all taxonomy groups for the selected family, class, or subclass. Okay, what I've presented so far is pretty standard functionality that you can find in many databases. In addition to that, we are trying to come up with new and exciting ways to visualize our data. One of such ways is the Sankey diagram. Sankey diagram is a flow diagram in which the width of the links is proportional to the flow rate. In our case, we display the links between foods, the first column of the diagram, compound subclasses, the second column, pathways, the third column, and disease, the fourth column. The entire diagram is pretty large, so I'll show you different parts of the diagram. In the center, you can see the highlighted link between grains and amino acids. The information panel for this link shows that all the composing compounds are from the family of organic acids and the class of carboxylic acid. Some of those compounds belong to the alpha amino acids and derivatives subclass group. Finally, there are a total of 6 compounds in the link between grains and amino acids, and all are displayed in the information panel. Users can click any of these compounds, compound family, class, or subclass to see more detailed information about them. On this slide, you can see the highlighted link between amino acids and metabolism pathways. This link is composed of a larger number of compounds, 15. We can see all of them in the information panel. Again, we see that they all belong to the family of organic acids and the class of carboxylic acid. They are split into several subclass groups: alpha amino acids and derivatives, and amino acids, peptides, and analogs. All these compounds are linked to various pathways, but all those pathways are related to the metabolism. Because of the large number of pathways, we display only the first tier pathways, such as metabolism, immune system, neuronal system, signal transduction pathways. We also display all pathways in the information panel. In this example, there are 35 pathways composing the link between amino acid and metabolism. The list of 35 pathways is not very readable. That's why we display the overview of these pathways and their relation to metabolism. To see this overview, users can click the button View Hierarchy in the information panel. On this slide, you can see the hierarchy of the pathways that is displayed after clicking the View Hierarchy button. In this hierarchy, the red pathways are the pathways directly linked to the amino acid compounds. The rest of the pathways show the hierarchy between those red pathways and their common parent, the metabolism pathway. Finally, on this slide, you can see an example of the link between the metabolism pathways and the cancer disease. There are a total of 119 pathways and 182 diseases composing this link. Users can see all these pathways and diseases in the information panel. Similar to the pathways, we display only the first-tier diseases, such as cancer, genetic disease, digestive system disease, etc., on the Sankey diagram. If users want to see how all 182 diseases are related to cancer, they can again click the button View Hierarchy. The purpose of the Sankey diagram is to allow users to explore relations between foods, compounds, pathways, and disease. To help with navigation and exploration of the relations, users can interact with the Sankey diagram to highlight specific relations. Finally, one issue that we are dealing with when building the Sankey diagram is the compound misrepresentation. For instance, according to the Sankey diagram, amino acids are presented in grapes, chocolate, onions, but not in citrus or apple. This is not because amino acids are not contained in citrus and apple, but because they are not measured in the corresponding studies or not linked to any pathways and diseases. It is hard to completely eliminate this issue because it's caused by incompleteness of the data that we can acquire. However, we'll work on improving the compound representation as follows: changing the algorithm to calculate width of the links, populating our datasets with more data, and adding a filter to the Sankey diagram so users could ignore certain compound subclasses. So far, I presented the current state of the MetaboFood web application. Of course, we still need to do quite a bit of work to make it fully functional. On this slide, you can see the tasks that we are planning to work on in the near future. First, we need to add more data from Dr. Colin Kay and Sierra to our application. We also need to finalize and test the interface. Once the interface is complete, we'll deploy it on the cloud servers and make it available to everyone. And of course, we'll work on more visualization tools to explore the data. Dr. Jin Yang has a few more ideas for data visualization that she is going to talk about next. Thank you.
Good morning. My name is Jin Yang. I'm a professor at the Computer Science Department of UNC Charlotte. I would like to introduce more visualizations that we may include in the web interface of the MetaboFood knowledge database. We prototyped them using Neo4j, Tableau, and D3 with real data from our knowledge database. In this talk, when I say a food, I refer to metabolome, which captures what's in the food or reported elevated post-consumption of this food. In the figure on the left, each bar is associated with a disease. The length of the bar represents the number of distinct triples of food, compound, and pathway to the disease. The diseases are sorted by length of the bars. It can be seen that the relationships between food and cancer are most widely sampled in our knowledge database. The figure on the right is a self-organizing map. Each cell represents a food. The distance between two cells indicates their similarity based on their compounds. This map shows an overview of foods available in the knowledge database and the relationships among them. The two views are coordinated. If I click a bar on the left visualization, the foods related to that disease will be highlighted on the right. Let me click the cancer bar. Both views are changed. The cancer bar is highlighted. The foods related to cancer are highlighted in the map. Let me click the Track Compound button to examine compounds of the selected foods. They are related to cancer. A loading diagram with 11 foods and 77 compounds is opened. The foods are represented by orange nodes. The compounds are represented by blue nodes. Allium, the rightmost orange node, has several compounds that are not linked to other foods. I clicked them so that we can read the names of those compounds. Now I want to focus on the relationships among the foods based on their cancer-related compounds. Let me reduce the size of the compound nodes. We can see that grains and cocoa chocolate share the multiple compounds. Berry and citrus also share the multiple compounds. We can see that allium share the 4 compounds with cocoa chocolate, share the 2 compounds with citrus, and also share the 1 compound with multiple other foods. Now let's take pathways into consideration. This node-link diagram shows 11 foods represented by orange nodes. There are 77 cancer-related compounds represented by blue nodes, and 213 pathways from these compounds to cancer represented by small red nodes. To distinguish links linking foods and compounds, and the links connecting compounds and pathways, I made the links connecting foods and compounds thicker. We can find many interesting patterns from this figure. For example, we can find that glutamate from allium has many pathways. Quinoline from grains also has many pathways, and there are 3 acids shared many common pathways. Next, I want to see if citrus and allium share any pathways in detail. I selected citrus and allium to create a new node-link diagram with those 2 foods and their cancer-related compounds and pathways. This node-link diagram shows the compounds and pathways from citrus and allium to cancer. We can see that they shared a few compounds that are related to cancer. They also have different compounds that share the same pathways to cancer. For example, glutamate from allium and pyruvic acid from citrus share the multiple common pathways. We can click the nodes to see their names. So one of them is serine biosynthesis. To focus on the shared pathways, I clicked a button to keep those pathways only and increased the size of the pathway nodes. Citric acid cycle captured my eyes. Why is allium also connected to this pathway? I zoomed in to check the details. It seems allium shared 2 compounds with citrus, which lead to the citric acid cycle. This is another prototype. The bar chart on the top left shows the counts of distinct triples of food, compound, and pathway to each disease. The bar chart on the top right shows the counts of distinct triples of compound, pathway, and disease related to each food. Cocoa chocolate has the biggest counts in our knowledge database. In the bottom view, compounds from each food are displayed along the x-axis. The compounds have 1 or more circles on them. The Y position of each circle represents the number of pathways from the compound to a disease. We can hover the mouse over the circles to check their details. For example, this is an acid in berry that has many pathways to different diseases. A component in allium captured our eyes. It's glutamate. It has 37 pathways to cancer. It also has 34 pathways to genetic disease. We zoom in into cocoa chocolate. Cocoa chocolate has multiple influential compounds to many diseases. Is it the best food for us? We need to be aware that there are much more publications on chocolate than foods such as a banana. We will make this information transparent and the publications accessible to our users. We can also filter the data by disease. After a disease is selected from the bar chart, the foods will be resorted by their triple counts on that disease only. The bottom view will only show the number of pathways from a compound to that disease. For cancer, we can see that onion ranked the third and berry ranked the fifth. While when we click mental disorder, we find that berry is more influential than onion. I have presented my two example visualization prototypes. We will evaluate those visualizations with target users before we implement them in the web application. I want to emphasize that in all the development stages of this project, we will always keep users in the loop. We will conduct interviews and surveys with our target users to learn what they want to do with the database, so that we can provide visualizations to allow them to conduct their tasks in the most straightforward way. We will also evaluate our designs and implementations with real users to make sure our visualizations are intuitive and effective to target users. In addition, since the information in the knowledge database is connected using automatic data mining approaches, there might be errors in the data. To improve data quality, we will not only allow users to temporarily or permanently block or change non-perfect data in their own view, but also provide a mechanism in the web application so that users communicate with the knowledge database manager if they spot any mistakes. The manager will then evaluate the issues carefully and make permanent changes to the database. Thank you for your attention.
So far, we have heard from Colin why we are developing MetaboFood KDB. We have heard from Sierra, Alex, and Jing the technical details in our implementation of the knowledge base. Next, I will place the knowledge base in a bigger context and provide a 10,000 feet bird's eye view of where we might take the knowledge base in the next few years. This is my disclosure. MetaboFood KDB currently connects dietary phytochemicals with diseases. It has a dedicated cloud platform that's developed following the FAIR principle to make the information in MetaboFood KDB findable, accessible, interoperable, and reusable. The knowledge base is also equipped with visual analytics tools for data exploration and knowledge discovery. All of these bioinformatics capabilities, together with other bioinformatics capabilities that our team has developed in the past, will allow us to develop a computational workflow for predicting the nutrition dark matter and further knowledge discovery. In addition, the vast amount of existing data scattered in various databases provides a solid and invaluable data source for researchers to apply artificial intelligence and big data analytics to the eventual design of precision diet. As Colin has described, the workflow for precision nutrition includes these roughly 7 interconnected steps. For the first 4 steps, Colin's laboratory has generated the evidence mapping data. The mapping data contains more than 2,000 compounds, and we expect it to grow continuously. For each compound, information on compound identifiers, taxonomy, and food source is provided. With the evidence mapping data from Colin as a starting point, our team has worked very closely to build the informatics infrastructure and the content of MetaboFood KDB. Sierra has written hundreds of lines of Python scripts to link phytochemicals to diseases by connecting HMDB ID to CAC compound ID to reactome ID to reactome enhanced ID to reactome pathway ID to CAC pathway ID, and finally to disease IDs. Alex has developed the cloud resource using SQLite, Flask, and D3. Jim has developed visual analytics tools using self-organizing maps, Sankey diagrams, node link diagrams, and Tableau to allow users to interact with the data. All of these efforts have made MetaboFood KDB a cloud platform for knowledge discovery with fun and ease. The development has been truly a cross-institution and cross-discipline collaborative efforts. The knowledge base will help us address the issue of nutrition dark matter. Nutrition dark matter are unknown metabolites. They are ubiquitous and numbered in hundreds to thousands. They appear in our bodies after eating, either directly from our food or in response to host and microbial metabolism. They show up on the molecular level as unknown mass spectrometry signatures. Our nutrition community needs to uncover their molecular identity, find out where they are from, and understand their functions for the eventual goal to prevent and treat diseases through precision nutrition. In this tentative computational workflow, we take advantage of MetaboFood KDB and the existing bioinformatics capabilities that our team has developed to facilitate the prediction of nutrition dark matter. First, untargeted mass spectrometry-based metabolomics data is acquired from human biospecimens such as blood and urine. Compound information is then extracted from the complex mass spectrometry data using the software tool ADP-Big. Extracted information then goes through ADP-KDB, a cloud resource for compound identification, annotation, and prioritization. Finally, machine learning algorithms are used to predict compound structures and identity. Information in MetaboFood KDB on nutrients and their metabolites, metabolic pathways, and diseases will be additional data that the machine learning algorithms could use, in addition to the mass spectrometry data. Among the existing bioinformatics capabilities in this computational workflow, ADP-Big is an open source, cross-platform, and graphical software tool. ADP-KDB is a cloud platform for tracking and prioritizing metabolites by taking advantage of the vast amount of metabolomics data in public data repositories. Recently, we have added algorithms for automated compound identification and annotation. One of the data sources of ADP-KDB is the Metabolomics Workbench. The Workbench currently hosts metabolomics data from about 2,000 studies. Among these, many studies focus on nutrition. Here I am showing none of them. Our team has been conducting a unified secondary preprocessing of raw mass spectrometry data in the Workbench. URLs are provided on the Workbench website to come to ADP-KDB to visualize the preprocessing results. We are currently in the process of also bringing in to ADP-KDB relevant data in GNPS, the Global Natural Products Social Molecular Networking. We hope information on natural products in GNPS will make ADP-KDB more useful for precision nutrition. With the prediction of nutrition dark matter, we hope that our MetaboFood KDB can get our research community one step closer to precision diet. What else could MetaboFood KDB do toward precision diet? First and foremost, we hope to bring the vast amount of data that have been collected through decades of painstaking efforts into a networked form to maximize their utility. These data are scattered in databases developed by USDA, NIH, and other research groups. This is a flagship USDA database, Food Data Central. The data are stored in JSON and CSV format in many files. For example, the data for the foundation foods are stored in these tables. From this record counts table, we can see the large volume of information contained in these tables. These tables contain identifiers to link all the tables together. For the USDA's Food and Nutrition Nutrient Database for Dietary Studies, FNDDS, food and ingredient information are also stored in multiple spreadsheets or Access and SAS databases. Data in these tables could be linked through the identifiers in each of the data tables. These identifiers include the food code, ingredient code, and FDC ID, Food Data Central ID. For the USDA's specific component datasets, we make a few clicks starting from this list to get to the tables for flavonoids. Here we can download the flavonoid data in Microsoft Access format. The data is stored in 10 tables in Microsoft Access with identifiers that could link all the tables together. For the NIH Dietary Supplement Ingredient Database DSID, the starting point is this web page. Clicking DSID will take us to the data files, and click on the data files will take us to the real data in Excel or Microsoft Access format. And this is the data in Microsoft Access format. Again, we can see many tables with identifiers that can link all the tables together. In addition to the USDA and NIH databases, there are many other databases that are invaluable for precision nutrition. These include the Phenol Explorer, a comprehensive database on polyphenol content in foods. PhytoHub, a database containing detailed information about dietary phytochemicals and their human and animal metabolites. FoodBee, a comprehensive resource on food constituents, chemistry, and biology. In addition to these databases, there are many other databases that I did not list here. All of these databases are truly gold mines that are awaiting bioinformaticians like us to bring them out of their individual comfortable homes and make them talk to each other. Eventually, we hope that our bioinformatics efforts on MetaboFood KDB will provide a comprehensive cloud resource for researchers to use artificial intelligence and big data analytics to make precision diet a reality. Thank you.
All right, that concludes our presentation. So if you can please continue to submit your questions in the Q&A area, and our panelists will be back visually in a moment so that we can pose the questions to them and hear their answers. All righty. Wonderful. Let's get started. Thank you, presenters, for those wonderful presentations about your very exciting work moving this field forward. Let's start with the first question. The current data feed is derived from existing databases. Is there a plan to have data inputted live from wearable devices by users?
Who wants to take that one? I guess I can start in that respect. We are really just in the infancy of this project, so we're starting with a highly curated data, so we know where it came from and how to expand it basically, and then we'll move out into less and less curated data. Moving on to something that you're speaking of that would allow us to actually pull, or users to pull data from say Fitbits or some other devices or apps. That would be a little down the line, but ultimately I think that's where we need to go if we're going to go into precision nutrition that helps users like people and not just researchers. So I don't know if Jean or any of the other panelists have ever worked on these types of concepts, but I know Shusha, you're working on some other diet-related approaches. You're muted, I think, Shusha.
Yes, we are indeed in the process of developing both wearable devices, and actually it's not wearable yet. It's a mobile device with a mobile app, and also to build a web platform to receive the data from the mobile devices. So eventually, we of course hope we could connect that mobile device connected web platform with our MetaboFood KDB. Then we will be able to incorporate data from mobile devices to our MetaboFood KDB. It is in our plan, but it's going to be a few years down the road because we're still developing the device, the biosensor, actually nanotechnology-based biosensor. We have developed the mobile app. We are developing the Bluetooth so that the mobile device could send the data to the mobile app. And so things are in our, they are currently ongoing and in our agenda. So that's a very good question. Thanks the listener for asking this question.
All righty, moving to the next question. How do you validate the compound pathway disease relationship? And another similar question, what data are being used to associate compounds with disease?
Did you want to take that, Sarah? I can add if necessary.
Yeah, so the validation or the information that's coming from the pathway to disease is used, the database uses CTD. So we actually just matched those IDs that we found from Reactome or KEGG and the ones that have already been collected and put into that expansive database.
Yeah, so as far as validating, we're kind of in the learning stages here because actually validating a lot of these phytochemical links, particularly, there's very weak relationships, right? So in a lot of cases, we are going to need researchers to use MetaboFood KDB to either generate a hypothesis or form specific study designs, basically, either in something like a nutrition intervention trial or to go into various cohort studies to see if these relationships hold up, basically. But for, like I say, for phytochemicals, the linkages are relatively weak because we just don't have the data. So we're basically piggybacking on endogenous metabolism and disease pathway data. For nutrients and vitamins, obviously the associations are a lot stronger. So I think, again, we're in the infancy here in precision nutrition when it comes to phytochemicals, and we really can't wait 100 years till all these associations are made, and that's why we're kind of developing this tool to give people an idea where we should be looking and what proofs we're going to need to take this further.
Maybe I can add a little bit. So as MetaboFood KDB grows, data curation, including the curation of not only compounds, Colin's lab has taken on tremendous efforts to do that, but we also need to spend tremendous efforts to curate the pathways, both metabolic pathways and disease pathways, because this will be a resource for people to use, and the data quality will be essential for the longevity of MetaboFood KDB.
Yeah, and to add to that, we do have tools at our disposal that will allow us to come up with more linkages between phytochemicals that are structurally similar to other things that we have stronger pathway links for. And that's something that Sierra, I know, is working on in the future.
We have a couple of questions about nutrition dark matter. The first one says, how do you evaluate the health and biological significance of nutrition dark matter? And similarly, another comment and question, fabulous, love the idea of nutrition dark matter. What specific dark matter should first be targeted? Also, has there been work on autoimmune diseases, like the examples given for cancer? Fabulous work.
Thank you. Maybe I can take up the question and other panelists could add. So for the second question, how do we select nutrition dark matters for prediction? We do have the compound prioritization stage before the compound prediction. Our ADP-KDB was actually originally developed specifically for the purpose of prioritizing unknown compounds. The idea behind that is to use publicly available metabolomics data to look at this particular unknown metabolic signature has appeared in what kind of studies, what diseases are studied, what sample sources are used. So those information was used in our ADP-KDB to provide us a quantitative measure for us to prioritize unknown compounds. The reason the prioritization is needed is because currently the mass spec instrument is truly wonderful. They could pick up so many signals. Among those, a substantial proportion of those signals are unknown. So we have to have a prioritization method so that the downstream compound identification efforts are not wasted, because really identify a compound is both costly and time consuming. So that's how we will prioritize the unknown compounds. And for the first question, Janet, do you mind repeating that so that I can target my answer?
Yes, so the first question was, how do you evaluate the health and biological significance of nutrition dark matter? And then carrying on, what was the specific dark matter? What specific dark matter should be targeted first? Right.
Okay, so to predict those nutrition dark matter, we have two sources of data to rely on. One is the mass spectrometry data together with chromatography information. The additional piece of information is the pathway information we have curated and the disease information we have curated. So the health benefit of the nutrition dark matter is already reflected in the metabolic pathways and the disease pathways. And those information we think will be important for us to predict the nutrition dark matter. I hope that answers the question.
Here's a short but important question. Excellent, I'm excited. When can the general public access it?
I'll let Alex answer that question.
Yeah, I guess different parts of the website now at different stages of development. for example, the stuff that I presented today, we can make it available probably in few weeks or in a month. And it will include just the search capabilities and the Sankey diagram where you can see the links between foods and compounds and pathway diseases. The idea that Dr. Jin Yang presented, I guess it's more in early development stage right now, so probably take more time to implement them. But I would say, yeah, we can make the website available in a few weeks or in a month, and then keep updates on it as it grows.
Well, I can add to that too. So basically, we're at the stage now of where we're able to interrogate it. And the nice thing about the data visualization is obviously it's hard to find issues or errors with data when you're looking at hundreds of data sets. But the data visualization is a way to look at all the data together. So we're really playing around and just trying to basically QA the data and make sure, is there anything that looks odd that we have to go back to the source data? And we have found a couple things. It's been really great for that. So we want to make sure that we release it when we're convinced that the data quality is there, and that's why we started with this really highly curated approach. So I think we need a couple of weeks to digest it ourselves, and then yeah, sometime in the new year, I would think early, it should be available.
Another question, is there a potential to look at dietary patterns as a whole rather than individual foods or food categories?
So when we get enough data in there, there would be. So again, we're starting with highly curated data, but we're also working with IFANS and the USDA in some of the upcoming expansions. We got 2 small library expansions, and then we're working with the polyphenols databases, and we hope to extend to Food Data Central and to ODS, like Shusha was talking about. Once we get more data in there, then there definitely will be the ability for people to upload foods in their diets and to look more at a total dietary knowledge base rather than just individual foods. But for now, until we get, as the data becomes richer, we'll have more and more capacity basically. So we're still building that in. Did you have anything else to add, Shusha?
No.
Okay, another comment. Thank you for the great work. Really enjoyed it. The topic is really comprehensive, but I was just wondering if you take into account any confounders in food items, for instance, chocolate, saturated fatty acids, or the consumption type, fried onions, or storage time. Let's see, it says that would have come from the same publication. So I think the question is about more, going a little deeper in food processing or any kind of confounders that may be a little deeper, like fried onions or storage time.
So let me just see if I understand this right. So you're talking about confounders in analytics from source data out there, or?
I think the question, because I'm a little unsure too, but I think the question is about confounders of, I think, additional information that maybe isn't, so storage time isn't necessarily included yet. You're concerned about that, or
Yeah,
I think that, or post-harvest. I think that the question is about that.
Yeah, I kind of get it. So this is more of the bioinformatics or big data analytics approach, where we're not so much focusing on the minutia, but on the big picture. So if there's anything that is reported for that food, regardless of it's only initially in the food or it's an artifact that comes from manufacturing, or if you're worried about degradation, we're initially concerned with, can we identify these structures in human blood samples or urine samples to be able to predict the impact of these compounds in cohort studies and things of that nature. So we're not really focusing on quantitative abundance, like I mentioned, or static levels of things. We're capturing what's in the food and what's reported in the metabolome. So we're basically capturing all compositional data regardless. Now, there will be issues with things like, we don't know in a lot of these studies if something has been annotated or identified. So was it just a mass that they found and they predicted the structure, or do they have a reference standard and stronger identification data? We won't know that level of detail basically, but what the goal is that the more data you have, the data visualization can represent the amount of data. So that gives you an idea of quality. If something's reported 1,000 times versus one time, that gives us further information. So that's kind of what we're getting at with the big data analytics approach. I don't know if anyone else had anything to add.
I can add a little bit, Colin. So we all know data processing, not data processing, food processing will affect nutrition value. Storage will affect nutrition value. So that level of data, we hope in the future maybe USDA or NIH could provide. We as a bioinformatics efforts, we take data and try to make it more usable. And the Collins group does produce data of your own. You'll have the analytical capabilities, but I'm not sure if you do that level of research to see how storage and food processing will affect the nutritional values. So to me, we rely on other organizations to provide that kind of data.
Yeah, and IFENS has really helped us kick off that initiative with the USDA. So we need to work with the experts, the USDA, Office of Dietary Supplements, CDC, whoever we need to work with that focuses on that level of detail. And we want to work with them and use their guidance along the way also. So this isn't just going to be a blind data dump from every data set on the internet. We're going to work with the experts in each of the fields that will make this data more usable. So people like the Food Data Central team, as an example.
Here's a question. If a researcher has a semi-quantitative metabolomics database, would that researcher be able to predict the direction of each pathway, increase or decrease?
Maybe I can tentatively answer the question. Other panelists could jump in. So our hope for MetaboFood KDB in the future is, after we have initial data, as much as we could to collect and put into MetaboFood KDB, we will have a foundation for us to develop artificial intelligence-based algorithms to help to not only to predict nutrition dark matter, but also to predict what kind of diseases might result from a combination of this diet. What kind of metabolic pathways will be activated based on this kind of diet that our people eat. But again, we are talking about a few years down the road for that type of work. We see there are many opportunities when we have this infrastructure built.
And I guess one of the researchers will be kind of able to do that initially on their own. So if you have a data set and you've identified a number of masses, and let's say you've got a list of formulas, you'll be able to upload that into our database, get some common data elements that you require to go to something like MetaboAnalyst or one of these other websites where you can input your intensity data from your output data sets and make those linkages a little stronger. So our goal is to allow people to, we're not replacing databases that are already out there. We're allowing people to connect with other databases. If it's beyond what we have, you'll be able to get the information, the common data elements or chemical identifiers or pathway identifiers that you need to explore your data in other platforms. So Even ADAP is another platform that we'll be heavily linked to.
Right. To further add to what Colin has just described, if users have mass or isotopic distribution, if you have mass spec data, you could upload your data to ADP KDB. And using ADP KDB, you can get a tentative identification or annotation of the data, and you know what kind of the compound could be, and then come to MetaboFood KDB. Eventually, we do hope we'll connect ADP KDB with MetaboFood KDB. They were designed for different purposes.
We're short on time and we haven't been able to answer all the questions, but let's definitely get to this one. Can you comment on the cost and the funding for such an effort?
I'll start and then she should finish because she's seen these through a lot further than me. We're doing this ourselves initially because we think this is important. So we've received really very little formal funding for this initiative. What we hope is we can get more funding to expand this and to take us to the finish line a lot faster. So Shusha, did you want to comment?
Just as Colin had just said, we are on very limited funding to do this. Most of us do this in our spare time. And so we do have a grant, a HERE grant, so we could piggyback on that because nutrition is considered part of the exposome. But beyond what HERE could fund, we have to seek for other sources of funding. Each of the panelists you see here has been very passionate in developing MetaboFood KDB, but sooner or later we need to find substantial funding to sustain these efforts.
So I'm so sorry that we haven't been able to answer all the questions, but we are out of time. So this concludes our webinar for today. Thank you so much to IFANS for hosting, our presenters for presenting this very exciting and future pushing work, and to our attendees for being here and for asking these insightful questions. Have a wonderful rest of your day.